Hi, everyone. Welcome to Foresight's Biotech and Health Extension Group. I'm really excited today to have the Biomarkers of Aging Consortium with us live to discuss the Biomarkers of Aging Challenge that is a recently launched open science initiative to really stimulate research on standardization, collection, and data gathering around biomarkers. Thank you so much for joining, you guys. Please take it away. The stage is yours, and I'm excited for questions.
Great. So I hope you can all see my screen. My name is Chiara. I'm part of the executive committee of the Biomarkers of Aging Consortium. And today, together with Albert and Seth, we'll be talking about the Biomarkers of Aging Challenge. And we really want to make this kind of interactive. So we'll have a demo later on and hope to answer all of your questions relating to the challenge. So we're already a bit delayed, so I'll just get right into it. And here's a brief outline of what we'll be talking about today. So I really wanted to give a quick background about why biomarkers. I think most of us on this call are probably quite familiar with it, but we really want to share our reasons why we're doing this challenge and what's major barriers for the field of biomarkers of aging at the moment. And then I'll briefly introduce ourselves a bit more, and then we'll talk about the 5 W's, what I've termed it. So why, when, what, who, and where. So all the details for the challenge. To go back to the basics a little bit, I thought I'd show this illustration and why we want to have biomarkers of aging. We know that we've really drastically extended lifespan of humans with modern medicine. And I'm from Austria originally, and we have a lifespan of approximately 80 years these days. But actually, if you look at the years that we spend in good health, then we see this is much less than our actual lifespan. And we have usually around 20 years with at least one age-related disease. And obviously in the field of longevity, we're interested in extending our lifespan, but what we really don't want is to push out these years with age-related diseases. Instead, what we want is to minimize the years with age-related disease. So we want to extend what we call health span, and this is a term that will come back later. But essentially, this is the years before the onset of age-related disease. And to do that, we might be able to do certain interventions. From research, we know now that many things can improve our health and prevent age-related disease, including dietary interventions, exercise, relieving stress, and so on. And these can be individual. So what works for me might not work for you. But what we really want is to measure the effectiveness of these measures and also test which interventions work. So this is a key challenge that we're having at the moment. So in humans, this research is quite challenging. So a lot of the work this far has been done in animal models, including mice, Drosophila, and so on, where you can easily perturb either genetics or pharmacological things. But in humans, this is quite difficult. First of all, paradoxically, we do have quite a long lifespan. Actually, to study an intervention and wait until people die or develop all of these diseases is relatively challenging. And what's often a challenge as well is to monitor the individual impact of such interventions. So really, did it work for me and for the person next to me as well? And this is one of the reasons why we would really like to have quantifiable measures of aging. And this is what we call biomarkers of aging. And as a consortium, we've recently come up with a consensus definition of what this would be. And that's essentially a biological measure that alone or in composite gives us more information about our health than chronological age alone. And the Wall Street Journal has illustrated it that way. So it's telling us a little bit more about what's going on inside our body. It may sound like this is something relatively new or fancy, but actually as early as 1969, this statement was made by Alex Comfort that kind of a test for human physiological aging is now long overdue. And here we are in 2024, and we still actually don't have any tools like this that are commonly used in the clinic. How can we apply these biomarkers of aging, or what could they be used for? At the very kind of basic level, we can use them to study molecular changes associated with aging and identify molecules that could modulate aging in a lab setting. They're also commonly used at this point in epidemiology. So what that means is basically we look at large cohorts of people or biobank samples, and we can quantify biomarkers and then how are they associated with disease risk. And this is one of the very big aspects of biomarkers of aging research at this point. And something that is a bit more forward-looking is really to use them for clinical research and personalized longevity medicine. So they could be applied to monitor over time how a person is aging and how they respond to different treatments. And this is why we think they're really one of the key components to unlocking longevity medicine. Those of you familiar with the field, and maybe even those that aren't so familiar, may know that these biomarkers have been developed, and there's a lot of papers, but actually not many or none so far are really used in the clinic. Why is that? First of all, there's a couple of challenges regarding characterization. So what biomarkers should we use under what circumstances? Validation. Do they really work for the purpose that we're intending them to use? How can we implement them in current healthcare settings? Most of us know that the current healthcare settings are very much focused on what we call sick care. So really people that are ill at the moment, and it's not really trying to monitor or optimize health. And what's also a major challenge is benchmarking. So we have publications that describe the performance of these biomarkers, but they're not really comparing using the same population. So we don't really know what is the performance of them if given the same population. So there's really relatively little standardization and consensus in the field, and this is something that we've been trying to address as the Biomarkers of Aging Consortium. I've already been talking about this a little bit, but I wanted to show some faces, who and where we are. So we were founded in 2022 by leading scientists in the field of biomarkers of aging. And I want to highlight Madi Mokri and Jesse Puganik, who have really been driving this alongside mentoring from our amazing scientific committee and also our advisors. Also, obviously, shout out to Alison and Dane, who are on the executive committee and making this work. Just very briefly, before I hand over to Albert, I wanted to show you what we've been doing outside of the challenge. One thing that I mentioned is that there is a lack of consensus and standardization, and this is really something that we wanted to drive forward. So we want to build consensus in the field and make it so we all speak the same language. Because if you ask 3 different aging researchers, what do you think aging is? Then you will get about 10 different answers and nobody will agree with each other. We're using a Delphi-based consensus model where we invited over 30 experts over the various works that we've published recently and asked them to come up with consensus definitions. And we've come up with frameworks for terminology, recommendations for validation and implementation. So this was our first paper published last year in Cell, and we also had one recently relating to the validation. All of these can be found on our website, agingconsortium.org, so you can download the PDFs if you're interested. We're also hosting scientific meetings because, in my opinion, and in our opinion, there's probably no better way to drive progress than bringing people together and really foster knowledge exchange. Our inaugural symposium at the Buck Institute was sold to capacity. Sorry, let me find the picture. So that was at the Buck Institute last year. And we're currently organizing the November 24 meeting at Harvard Medical School. The sign-up is open, and you can also submit abstracts. A few of those abstracts will be selected for oral presentations, so really great opportunity to present in front of a leading community. And lastly, this is really what we're going to talk about today, is community building, innovation, and open source tools, not tools. So I'll hand over to Albert in a second, but I really wanted to highlight that this is something I think is quite innovative. Also, the challenge has been inspired by data science challenges, and Vadim Gladyshev gave us the idea that this could be something that would be very helpful for the aging biomarker field. With that, I'll hand over to Albert.
Okay, so today I'm going to introduce you the recent update on the BioLearn. Thank you, Kirara, for the introduction. The purpose of BioLearn is basically we're trying to tackle a problem of not standardized biomarker and data set. We really want to create a go-to tool for the biologists or data scientists to use for either building new biomarker for aging or applying biomarker for aging across different kind of cohort. we have made a lot of progress since our first release. Today we'll introduce our recent update basically on these 4 topics. So the data preprocessing, the cellular deconvolution, systematic clock assessment, and the survival analysis. So the first is that we introduced some new methods for people to do the quality control of data set. And so basically there's a very simple command. You can get a very complete quality control on how many % of data are missing, how many sample are outlier, etc. And we also have introduced this imputation method. Basically, also very simple. We have multiple different imputation methods available, and the simplest one is basically just impute from the reference data set. So we also show that by doing the imputation, compare with the data set with a lot of missing value, the imputation really can improve the quality of the prediction of the biomarker. And we also have a very new function with the help of we are able to let people to do this cellular deconvolution on the blood samples. Basically, we introduced I think 2 different methods. One for the decay array, another for the epic array. Basically, it allows you to, if you have a blood data set, and you can very simply apply this one-line function, and you can get the predicted deconvolution of the cell type composition. So basically, here's one example. This is on a data set that knows the real ground truth cell composition. With solution method, you can really just predict this cell proportion based on the bulk blood DNA methylation data. And what is really exciting is what this tool really enables is basically a systematic clock assessment. What I mean is that basically you can compare a large amount of clock biomarkers side by side. Here, basically, we apply the clock. Here, we're showing 5. Apply this to this NAS cohort. You can see each of the clock, they really correlate well with chronological age with different accuracy or correlation. If you can really rank them, here I'm showing case the 16 most popular or well-known clocks. You can see you can really rank them based on their correlation with age in terms of their performance in predicting chronological age. And what is more exciting is that we can even perform this lifespan analysis to basically look at how this clock associates with mortality. Correct me if I'm wrong, but I think this is the first time we ever show like all these 16 different popular aging clock together to compare them, like together their association with mortality. So here you can see the top ranking clock basically are, not surprising, the Greenwich version 2 is the currently most potent clock that predict mortality, and followed by Dunning Pace, and followed by Greenwich version 1, and followed by the damage, which is a causality informed clock we published earlier this year. So this is basically this tool really enable us to just effortlessly perform this large scale comparison across all the biomarkers. And this list can even grow larger in the future. And we also have this blood-based biomarker. So the phenotypic age, which is based on the blood count. So basically, you can also, if you have this kind of data set, you can also apply to this, take it to the BioLearn package and make the analysis. And what is more exciting is that we're expanding our universe of the biomarkers. So before I'm showing the 16 biomarker of aging, and now we expanded to almost like 33 harmonized biomarkers. So we're now including even more trait predictors. So we have this common trait predictor like smoking status, BMI, alcohol consumption. We also have this common disease predictor included like Alzheimer's disease, coronary heart disease, etc. And I would expect that in the future, we will be able to include, and also with the help with community, we'll be able to incorporate more and more biomarker, and even not just epigenetic biomarker, maybe also other based from also other omics. With this, I think, so you can, if you are interested, you can check our updated manuscript on bioRxiv. So it's already, and you can check detailed method and the code and on biomarker. And with this, I think I will pass to Seth. So he will show you a live demo on how to use BioLearn and how do you use BioLearn to submit to the challenge. Yeah, thank you.
Hello, everyone. Can you hear me?
Yep.
Cool.
Let's just share screen. Okay. All right. This doesn't let me share a window, so I'm going to have to do a lot of switching for the demo, but we'll see how it works out. All right. Biomarkers of Aging Challenge. Pretty excited about this. It's been in the works for a while. We're going to talk a little bit about what it is, and how you can get involved, but also wanted to, before we do that, just say thank you to Methuselah and Volo, especially for providing funds so we could have a prize for this challenge. A lot of other people have contributed. So why are we doing this challenge? We've seen success with these kinds of challenges before. Kaggle has become a household name, at least in the data science community, for its success in running these kinds of challenges. And a lot of companies have actually used this model to improve some of their AI models. So we thought it would be good to try this with biomarkers. We also thought this would be helpful to build engagement with the community. People who work on biomarkers is a relatively small subset. And we think it would be great if we got more people to look at this because it's a really important problem, biomarkers of aging. So get some people who aren't currently working on biomarkers to give it a shot. And also more people with a computational background. So more people with machine learning, data science type backgrounds. We think more of that could really help, especially with all of the omics data that we're seeing. We also thought it would be really useful to benchmark different approaches. So we could have kind of an open and fair competition of these different biomarkers to see which ones perform the best on the same data set. A lot of times these are developed on disparate data sets, and so it's hard to compare apples to apples. So what are we actually doing? We have an open competition that's happening. It's broken up into 3 phases, which represent the different things that we are trying to predict. So the first phase is chronological age. It's happening right now, but there's still plenty of time to join because there's 2 more months before we close submissions. This QR code on the side will give you a link to join, and that'll stay up through the rest of my presentation. Phase 2 will be focused on mortality, and phase 3 will be multi-morbidity. Phase 2 is going to be towards the end of this year, and phase 3 will be launching sometime in 2025. We're really excited about these. I think mortality and morbidity are really exciting outcomes if we can have good predictive models for them. For the competition, we have this data set of 500 individuals from the MGB Biobank. I think this is really shaping up to be a unique data set. Currently, we have EPIC V2 methylation data for these individuals. And that's special thanks to Fiona, Vadim, and Foxo for providing this data to us. And we're getting other omics data also, proteomics and glycomics, which we will be sharing our partners and how that's coming about soon. And we're also in talks to potentially get even more data on the same set. And once we're done with the challenge, our plan is to make this entire data set publicly available so it can continue to be used for study and for benchmarking of different biomarkers. We also have mortality and clinical data for all of these individuals. So that'll be useful for phase 2 and phase 3 of the challenge. So who should be involved in this challenge? You. If you have experience in biology or omics or data science, machine learning, writing code, any one of these things, you don't necessarily have to have them all. And if you're interested in working on producing better biomarkers of aging, then you should consider joining and join a team. So how would you do that? Where do you go? Here's our challenge home. If you go there, you can sign up right now. We also have a community Discord. I just created a new channel looking for teams. So if you want to advertise that you're looking for a team, just post there and say who you are and what your skill set is and what you're looking for. And maybe we can get some new teams. And also we have the BioLearn library. So this is going to have a lot of helpful tools for creating submissions. And we're going to go over that in a little bit. So these are the 3. I'm going to leave that up there for just a second. All right. So we're going to do a live demo. Fingers crossed. Hope this works. I'm going to sign up for the competition. I'm going to create a submission using BioLearn and I'm going to submit it. Here we go. So this is a new account. I don't have any access to any projects. So this is just like what you would see. I have done one thing, which is Synapse asked you to go through a little quiz before you can access any data. I didn't want everyone to have to see me do a quiz online. So I did do that part. But if we go to synapse.org/biomarkers, this will take us to the challenge where you can read all the details that you might want to know. And here we have a button we can click to register. Click continue, and I'm now joined to the challenge. Great. So how do we move forward here? We need access to data. So we go to data, And here we have, this is the age prediction challenge, and we want access to the data. So we click here, but it looks like I don't have access yet. All right. If we go to files, we have some data access instructions here. And there's a little lock icon. So I'm going to click there and I'm going to request access. It's going to let me go through this page and say, I want to request access, put in some information, and then I'm going to add the people that I want to be on my team. I'm going to download this PDF, fill it in, sign it, and then save it. And then I will upload it here. And once you've done that, by the way, this is a little scroll bar. This is maybe a little confusing, but this is a scroll bar. So once you've done that, you upload it. And once you upload it, within a couple days, you should expect to be approved. So I'm not going to do that process, but I do have access to the data already. So we're going to jump over to BioLearn and work on producing a submission. All right, so here is BioLearn, our documentation. We have some examples, one of which is here, loading up data for the competition and creating a submission. We also have one where it trains an elastic net model, a little more complicated. We're going to do the simple one for this demo. So I'm just going to download this Jupyter notebook. If you're familiar with Jupyter, it's just a way to interactively run Python. All right, so I've got that downloaded. So I'm going to do this, the developer version. Go over here to the repo, BioLearn, pull it down. All right, so I downloaded the code here. Yes, I trust the authors. So I'm going to open up a terminal here. I can't switch back and forth very easily. All right, I won't be able to show you the instructions, but I'm pulling the instructions from the site. Just says create a virtual environment and then install all the dependencies. It's going to take a minute or two to install all of these. Should be pretty fast. So I think I'm going to stop share on this. Actually, we're going to go back to the web browser. This is going to be easier. Okay, here we are back in the web browser. Everyone can see that. These are the instructions I was going through. So the install should finish up in a second. And now I will open up Jupyter. Here I have Jupyter. And I will take that notebook that I downloaded. Here it is. All right. And I will put in, so I already accessed the data using the method that I demonstrated. So I'm going to load up that data. This will take a minute. And let's pick a clock. Which clock do we want to use for this? Maybe Horvath? Anyone have any opinions? We'll put it in the chat.
Getting Pheno.
Pheno. I heard of Pheno. Okay, let's try Pheno. So Dunedain Pace does not predict age. It predicts aging rate. So it will give us not very good answers if we're trying to predict the chronological age. Okay, so let's see. I can't see. Okay, there we go. All right. So we've got our data loaded up. We're going to do Pheno age. Run that model. We'll call this the Pheno age submission. We'll kick that off. So this is going to take, actually, I don't know how long it's going to take. I haven't run Pheno age on this data. It's a pretty big data set because the Epic V2 has a lot of samples in it. And it runs pretty fast. All right, so we've got all our samples here. And now we've got it correctly formatted into a CSV that will be accepted by a challenge. Here's our CSV. Yep. All happy? Okay, so how do we actually submit this? Let's go back to the wiki. How to participate, submission tutorial. Okay, in the left sidebar, click on the projects icon and create a new project. Let's see. Duplicate this. Go here. Create a new project. Easy enough. Give it a good name. Build our model. We did that already. There's also a sample submission file here, so you can go through this process even if you haven't yet gotten access to the data to make sure that got it all down. And then we're going to upload our prediction. So we're going to upload a file. So I'm going to go to Files, upload a file, and then I will get that file. Here's the file that I created. Here it is. We can see it. All the data. All right, easy enough. Now I will submit it to the challenge. find the file. Success. And in a minute, it will get graded and it should show up here on our dashboard. Nope, on our leaderboard. There we go. So the leaderboard is going to show the top submissions. I don't know if we'll make it in the top 20 actually, but this is showing all the top submissions. So just for everyone's information for the judging for this competition, the leaderboard does not necessarily represent the winners at the end of this. The leaderboard uses half of the data to give you an idea of how good your submission was. At the end of the submission period, we will take the top submissions and grade them against all 500 samples. And if some scores are close enough within a very small amount, about a week in chronological time, then the tiebreaker will be the number of features. So less features will be given preference. All right, so that's everything. Hello again. People have questions?
Yes, we've collected a ton of questions in the chat already.
I haven't been looking at the chat much.
It's okay. I've been monitoring. We had Rune, Steve Fawkes, and a few others. If you still want to upvote questions, feel free to do so in the chat. We'll first go through the chat and upvote the questions there first, and then we go through raised hands. I know, Varun, that you had asked one quite a while ago, and if you want to repeat it here, it's about imputation. I'm not sure if it's relevant anymore. Otherwise, we can also skip to the next one.
No, I think, first of all, kudos to you all because this is a lot of incredible work. I think it might have been answered, but just to confirm, Albert, for the imputation method, it seems like it's a population mean based off of a reference data set. Am I understanding that correctly?
So the current implementation is the population mean. So you can define what data set is the gold standard by your definition. And
Got it. Okay.
So in the future, we may implement the KNN-based or more sophisticated method.
So there is an ability to select which kind of goes into that next part of the question, which is selecting which tissue type that you can actually impute from.
Yes. So you can define your imputation source. Yes.
Awesome. Okay, cool. Thank you.
We also have in the library right now, it's not as well documented as it probably should be, but there is a variety of imputation methods you can choose from. When you get a model from the library, you can specify what imputation method you want it to use. There's the option you can choose to pick the average from the current data set you're working with. You can choose to populate it from a reference data set like you were talking about. And there's also a hybrid methodology based on what Dunedin Pace uses, where if there's 80% or more of the samples are present, it will use the average. And if there's more than that is missing, it will replace everything from the sample mean for that entire CPG site. Few different options, and more can be created pretty easily if we want more options for imputation. That's definitely possible.
Follow up with from that, which that's awesome. First of all, is there any kind of effort going into a genomic-based imputation where you're using neighboring CPG sites as a way to stabilize for an imputation, or is that not on the horizon?
I don't know about that from a technical standpoint. I don't know if anyone in the group knows about that from a scientific investigation standpoint. If you wanted to help get something like that into the library, if you know how to do that, then I'd be happy to feel free to reach out and we can work on adding that.
Cool. Yeah. Thank you.
We have so many questions. We're going to try to rush through all of them. So next one, you have Abdul Calder. You have a question both in the chat and raised hand. So go for it. And please keep it to one for now.
Yes, the first question was about when you have a progression of a chronic disease, that you have population where you have fast progressors, these normal progressors. Can we say that some of those have intrinsically fast progression in aging as a cause, or is it not studied enough? That was the first question. The second question is whether these biomarkers for human apply to animal models, like, of course, primates and then lower mammals. How much can we extrapolate the success and efficacy of these predictors into the animal models? Those are the two questions. Thank you.
Maybe I'll briefly take this one. So both of those are really interesting questions. I think there has been some research into progression of disease and aging biomarkers. So there is literature on that. For the biomarkers of aging and mammalian conservation, I would refer you to really exciting work done by Steve Horvath as part of the Mammalian Methylation Consortium. I think they recently published a paper where they basically built a pan-mammalian biomarker that can predict the age of any species, and really exciting work. So yeah, definitely refer you to that.
Thank you.
Awesome. Cool. We have a long list still. Steve, you go next. I know you have a few questions for everyone. Steve Fowkes, please maybe keep it to one or something.
I guess my main question has to do with this focus on suites of biomarkers as aging predictors, as opposed to examining the merits of individual biomarker tests as might be added to such suites. And one of the ones that I'm aware of is the issue of cellular hydration. This is strongly associated with the developmental process and with aging itself and with pathologies of aging, end-of-life pathologies. And Emmanuel Ravisi proposed that this was a biomarker of cellular hydration, this potassium utilization issue, where you compare serum potassium to either total blood potassium or cellular potassium in blood cells as a way of looking at hydration. And these things, he said, tracked, and he published this without data back in 1961, but developed it 100 years ago in his clinic in New York City. And given the fact that we've got at least the neuroendocrine theory of aging proposes that the aging process is a remnant of the developmental process, and that hydration is associated with that we should be actually paying attention to that. And I'm not aware of any suite that includes either hydration or potassium utilization in their.
Yeah, that's a really good point. I think, so we're obviously interested in all sorts of biomarkers. Specifically for this challenge, we're focusing on omics and methylation. We're adding more omics later on. But I think, yeah, the hydration aspect is definitely something very interesting. I think there are some biomarkers that include serum like iron levels. Not aware of anything specific there, but yeah, I think it would be great to look at this in the future.
Yeah, the serum test correlates poorly with age, but the ratio of cellular potassium to serum potassium, the utilization of potassium, Ravisi said that correlates well, and he documented it in the context of cancer, where there were potassium overutilization kinds of cancers and underutilization kinds of cancers, and he would track these with a simple test of comparing serum potassium test, which is incredibly inexpensive, to total blood potassium, which is equally inexpensive. And if you wanted to do the math, you can subtract the serum concentration from the total blood concentration if you want to get that precise. But because 90% of the potassium is inside of cells, that's really not necessary. But this is a $10 test. So clinically, it makes a lot of sense to include it.
Wonderful. Thanks, Steve. Next one up, we have Matt, meaning Matt Soldano here, who asked a question in the chat.
Yeah, sorry. So for the tiebreaker, you were talking about using fewer features as opposed to more features. And I was curious why you would choose to go with fewer features, because just trying to think about this logically or in a biological sense, I feel like you want more and more features. Obviously, we're limited by computing power and cost, but at the end of the day, having more and more features would allow us to capture more differences and different situations for different people. And obviously, like, accumulating the multi-omic models, we're getting more and more features that are able to allow us to predict better. And so I feel like it should be more the other way around, trying to get more features.
Yeah, I can answer that. So this tiebreaker only comes into play if you're essentially getting the same signal. So you're able to predict just as well as the other model. And in that case, the more features is not adding anything. It's not getting better signal. And so fewer features for that reason is better for a potential assay in the future on a cost perspective. So an accessibility or portability perspective, because if you just need to measure 5 things, that's a lot easier than having to do these broad-based assays every time. If you can make a custom panel, the smaller that custom panel is, the more economical it will be to provide to people.
Awesome. We're breezing through them here. Next one up, we have Herbert.
Hi, thank you. I was wondering if the competition will start to include other types of methods or data sets that haven't been used before. There could be people out there who are using proteomics approaches or imaging approaches, just like things like MRI or something like that. I personally am working on developing biomarkers for aging as well. And the data set that I'm collecting is not listed within the challenge. So I'm wondering, will there be a way for people who are doing something outside of the scope as the challenge is defined currently to be able to still participate somehow? Thank you.
So we do have proteomic data and glycomic data coming. We have partners who are going to be producing that. I don't believe it's going to be ready for the age prediction part of the challenge, but we're hoping it'll be ready for phase 2. So there is that. We are limited, and I'm not the best person to speak to this, but we are limited for the samples that we have, the individuals that we have serum to do testing on. But we can't necessarily go and get imaging data for these individuals retroactively. There are some limits to what data we can generate for this set.
Cool. Stay tuned. Next one up, we have Nir.
First of all, thank you very much. This is, I'm on the SAB, but I think this is a great program. And I thank you guys for spreading the gospels here and getting people excited. I just didn't understand the cohort. You're taking a cohort of 500 people. And I want to make sure that you know that at least from proteomic and metabolomic, there's a very different biomarkers or extent of changing biomarkers after and before the age of 50. Actually, in 65, it's more. So I just wanted to know, what is this cohort? What is the age? And what are the consideration for the fact that there's different biomarkers? And I would add to that also means that this cohort of 500 is not going to be the same cohort for mortality and other stuff.
Seth, you're on mute, sorry.
If you go to our Synapse Challenge website, you can see we have distributions for the 500 individuals in the sample. So we've got a fairly even age distribution between 20s and 80, 90s. And then there's some drop off on the very young and very old. I'm not sure I understood the part of your question saying that this isn't the same cohort for the mortality. The plan is that this will be the same cohort for the mortality. So we have some percentage that have already died in this cohort.
It looks like you have 500, so you have 20 that died or something. I'm not sure that's a powerful cohort from that perspective.
It's fair. We'd like to have more.
Yeah, I think the strength of this data set, at least, is that this is brand new, so nobody's really touched it for training. And this really allows us to benchmark, at least for the existing biomarkers and any new ones, what is the mortality. But obviously, I agree that more data would be really great. So hopefully in the future, we can get more funding to expand our data sets.
I also just want to say that at the end, those 3 goals are great. At the end, we want to have biomarkers that we know are changing if we implement lifestyle changes or medication or supplements. And this is another complexity because not all biomarkers are changing or not changing in close enough time to show to the individual and in clinical studies that we're actually making a change.
Awesome. We have Gian next, and then technically we have John, Larry, and I'm not sure if anyone else has questions. I'm happy to stay maybe 5 minutes longer in case anyone else can, but unless you guys can make it, there's maybe no point, but perhaps we'll just see how much we'll
Before we move to the next question, let me just break in. If you don't get your question answered or you come up with a question later, the Synapse has a forum. And Discord also has a section for challenge support. If you want to post questions there, we'll definitely get to them and respond to you to the best of our ability. I will post the link to Discord in the chat. I don't know if it's up there yet.
It's all the way buried under the flurry of conversations. Next one up, we have Gian. All right, then maybe Josh Middledorf. Do you want to ask your question?
Yes, please. Thanks for recognizing me. So I have a really basic point about the way these biomarkers are being evaluated. I wrote a paper about it, which I know I sent to Albert recently at his request. So I hope it's on your radar. It's a really basic point. It's hard to get that on first hearing, but here it is as succinctly as I can present it. Suppose you have epigenetic response to damage. The body realizes, oh my God, we're really damaged and we need to ramp up our repairs. This is expected to be increased later in life, it's a biomarker of aging, but we don't want to set that one back. If we set that back, the body is actually going to have a shorter lifespan. So this is an argument that we need not statistics, but we need basic theory in order to evaluate, to filter which biomarkers should be included in our set. And there are some that should be and some that shouldn't be. It takes theory as well as statistics, is what I'm arguing. Thanks.
Yeah, thanks. Very good point. I think actually Albert has written a really nice paper about the aspects of what is really the damage part, what is our body adapting to aging. So I can definitely recommend that. But yeah, for this point of the challenge, we're really just looking for a biomarker of chronological age as accurate as possible. And actually, this will actually not be that helpful for any sort of biological interventions because it correlates really well with chronological age. And if you ask someone for their age, then you probably get the same information. But I hope we can build on the model, and then improve it to, develop predictors of mortality and ultimately health span, which is the most interesting part of it.
Yeah, it's even worse than that. You can have a really good predictor of mortality, but it turns out to have markers in it that are responses to the terrible damage that the body is experiencing. They're responses to the damage. They're not indicators of the damage directly. And if we're looking at epigenetic markers, for example, of smoking, we're bound to find such markers. Smoking shortens lifespan. If there's an epigenetic response to smoking, then it's probably about rescue. It's probably not about the body destroying itself or about something going wrong with the body. So those are the markers that really should be counted in the other direction in an aging clock that evaluates interventions.
I think we are in agreement that separating out the adaptive responses from things that are damaged is very important. And that requires kind of new thinking about how to approach these biomarkers so that we can get to, what everyone is really hoping for, which is a surrogate endpoint for therapies.
Thank you. I feel heard.
Wonderful. That's wonderful, Josh. Next one up, we have Larry, if you want to unmute.
Yeah, I think it's a good effort to predict chronological age, but I often find that a lot of times you learn the most from outliers, like people who look like, or your tests look like they're very old, but they're still very healthy. And I think, having biomarkers of health or future health is really what you may need, and have a living biobank in a sense that you have people that you can go back to. And so when you see something to further test, then I think a lot of it, I would agree with Josh, that a lot of stuff is due to damage. And even for smoking, even people who smoke a lot of cigarettes, which is terrible, many of them don't get lung cancer. Only 25% of those people get lung cancer. And why the others don't or something is an interesting question. Not that you want to promote smoking, and there's a lot of other things they get. They get emphysema, they get heart things and stuff like that, why certain organs are affected and others aren't. But I think looking at outliers is, I think, really a lot of times you learn a lot more. And this challenge may be biased against outliers or looking at outliers.
Yeah, I think the scoring may bias in some way against outliers. I agree with that. However, I think just for the sake of this challenge, we had to choose something. And I think what we're using is now very commonly used in the literature. But obviously, a very good point. And I think all of the discussions that we're having here as well maybe can also help people refine their biomarkers. So I'm really glad we're having this discussion.
Awesome.
I think some of that will be captured in phase 2 and phase 3, where we're looking even more at mortality and morbidity, because the people who smoke and then don't die, that'll be captured in.
Yeah, and health is such a stochastic process. You get a stroke one day, and if you could predict strokes, high blood pressure certainly correlates with it.
Sure.
But it's such a stochastic process. You get thrombosis somewhere in your leg, you're flying a damn plane, and you get a stroke the next day. And there's so many things like that happen in life. And so it's always very difficult to tell people how to prevent death.
Yeah, not to get too far into that, but I think heart disease issues are not just, they may be harder to capture with omics because in many ways they're partly anatomical, which is probably going to be the hardest thing to identify with a blood test.
and fibrillation or something. I don't know if you're going to have some of the people wearing wearables. You can pick up fibrillations and things like that, which sometimes are very, to prevent them is very important.
All right, we have 3 more questions here. Kevin, if you're still here, I know that you're about to head out, but you're welcome to make your comment. Now.
Yeah, it wasn't really a question, but it's just about the element of time as a variable. Obviously, 100,000 people dying every day is a pretty urgent thing, and there's more people heading that direction, including me and everybody I care about. So I'm interested in time. How do we introduce time as a variable that we reward people for minimizing in these competitions? And obviously, if you've only got one competitor, there's nobody really buying for first place, basically. But if you have hundreds, maybe, or even more, and they all start, and when does the information that they have, when is it potentially useful to others before it's fully baked? Maybe there is a reward, a time element that you can reward people for releasing information before they feel it's completely ready or somehow. So how do you reward transparency and speed? And first of all, you have to have enough energy or enough competitors to make that variable worthwhile. So that was just a comment.
Maybe I can start with that and then hand over to Seth. Maybe for the time aspect, I think is really interesting, but we're also a little bit bound by regulatory aspects, especially anything to do with medicine. So this is obviously one of the issues. In terms of the transparency, though, basically for the submissions and especially for the finalists, we will ask them to share their code with us and really provide a reproducible example, because too many times in the literature, there's something where you ask the authors and you never get a response, or you get really badly annotated code. So we really wanted to make sure of that. And maybe I'll hand over to Seth, pointing towards the rules. But yeah, we're really trying to make it as transparent as possible.
Yeah. So time, if you mean time to how do we get these things into affecting human health, that is a hard problem. And as Chiara said, I think regulations are probably the biggest obstacle to moving forward on that quickly. But certainly we still need just good things if we want to use them on humans. As far as transparency, as Chiara said, we will be looking at code and the models and how they were trained. We're not requiring participants to disclose to disclose their code, to share it, for it to be open source. But we will be requiring the winners, at least, to share their methodology. However you did it, your code, we're going to look at that to validate that you didn't cheat or something. But then there will be a description that will be shared with everyone, so these ideas can be propagated in the community. And we'd like to be more transparent. We would like to say we only use public data sets, but especially for mortality, morbidity, there's just not enough data available as far as training. So it's really hard.
Yeah, no, this is the beginning of a revolution, Seth, really is what it is.
We've got a lot of work to do.
A lot of work to do. And I think AI, we haven't talked about that. It hasn't been brought up, but that's another big, huge elephant, is how does AI fit into this now that it's actually becoming available? Anyway, I'll leave that for another time.
Happy to talk about this in the future if you have ideas for how it can be more transparent or move faster.
Cool. Ethan, do you want to get your question in? I thought it's relatively simple. Otherwise, I'm happy to ask. It's, are there considerations of digital biomarkers data in measuring aging?
Sorry, what was the question?
Are there considerations of digital biomarkers data in measuring aging?
Digital biomarkers are definitely biomarkers. They're interesting. There's some models that are out there. I know that at least one company that has one. So I think continuous measurements like digital biomarkers are potentially very valuable, especially for the future. I would personally, I'm just speaking for myself right now, but personally, I'm very interested in this idea that if we can use omics biomarkers, which are relatively expensive and hard to acquire, to figure out some relationship with continuously measured biomarkers so that your phone or your smartwatch can tell you, hey, here's an anomaly that might match with this issue, go get a blood test, basically. I think that would be pretty amazing.
Okay, cool. Jason, you have the final question, and then maybe Seth and Chiara, if you, Albert too, if you feel so inclined to just summarize next steps or how people can get involved. I know the Discord is now at various places here, but it would be useful to maybe end there. Okay, Jason, last question.
Thanks, Allison. Just a quick question touching on what Josh was saying. I thought it was some good points. And I was wondering if this challenge is going to help to decipher between the passengers and drivers of aging and age-related damage, and speaking to the adaptations of the body. Anything here help us drive insights into that? I'm just curious if there was any thoughts on that.
I think I can maybe answer this one. So we have early this year, we have a paper on nature aging, just trying to tackle the question, can we separate age-related damage and adaptation? Basically, with this, it's an innovative approach. Basically, we take this causal inference approach into the clock model and use that to segregate the age-related damage and adaptation. So this is definitely one way to go. I think in the future, so if you remember the previous mortality association plot I show, the damage actually one of the top clock that predict mortality, although it's never been trained on mortality data. And the adaptage, the model that builds on adaptive change, there also is actually the kind of the worst clock they predict. They have inverse relationship with mortality. I think it's a very hard question whether how do we define damage adaptation, because we don't really have a gold standard to train on. So it's still in a hypothesis stage. But I think in the future, I think there will be more and more biomarkers that recognize this problem and trying to tackle it.
Gotcha. Thank you. Appreciate that.
And just to answer your bit in relation to the challenge, and specifically, one of the things you must have for a challenge like this is objective scoring criteria that everyone can agree on. I think what Albert has been working on, I think it's really cool that we're starting to tease these things apart. But I don't know that we could find yet objective criteria that everyone would agree on for judging a challenge. So I don't think the challenge is going to help too much on that particular question at this time.
Thanks, Seth. Appreciate it.
Okay, wonderful. Do you want to leave us with any final words, Seth, Kira, Albert? If people have forgotten everything, what is the one thing that they should remember?
BioLearn facilitates systematic assessment of the biomarker and easy application. So yeah.
Join our challenge. We want you.
Awesome. We have the Discord here in the chat. I'll also make sure that we'll include it, maybe even on the YouTube link afterwards when it's done and ready to be published. Thank you so much. This was quite a wild ride. We've had a ton of people joining, one of our busiest seminars in a while. Thank you everyone for your fantastic questions. Thank you so much for staying a little bit longer. And really excited about this. And I think we're just getting started. And we're really looking forward to all the applications. Thanks everyone. And we hope to have you guys on very soon to just share a little bit more about progress and what's happening and how people can join perhaps the next round. Thanks everyone. Have a good one.